This week, we will discuss mechanisms that viruses use to enter target cells. We will start with steps in virus entry, penetration through cellular membranes, and in second recording, we will discuss intracellular transport and replication and expression of viral genome. We know that viruses first must bind to target cell receptors. This applies as a first step, which leads afterwards to entry and penetration. We already mentioned that eukaryotic viruses can enter either by fusion, if they are enveloped viruses, or via endocytosis. In exceptional cases, they might trigger or drill the tunnel through cell membrane. However, the completely different way of entry is in bacteriophages and plant viruses. Why it is? Because bacteria and plants have cell walls, and therefore virus cannot neither fuse nor endocytose. So how do they enter? Here is an example of T7 bacteriophage. So once they attach to the surface of bacteria, this attachment triggered conformational changes in some of their proteins, which insert some cell into the bacterial wall and drill the tunnel through which afterwards the bacteriophage DNA is released into the target cells. For plants, in most cases, the viral entry is enabled by some mechanical damage, scarification, or insect can introduce a virus into the plant. Once the virus enters plant cells, it can disperse or infect neighboring cells. It can move via plasmodesmata. Now, envelope viruses, we mentioned already that they can enter by fusion of their envelope with target cells, while unenveloped and some envelope viruses enter by endocytosis, receptor-mediated endocytosis. So if you look on the right side of this picture, that is the entry of the virus into target cells. And we discussed before the budding and release of variant, and you can see it's more or less opposite mechanism than exit of cell. Some viruses exit by exocytosis and they might enter by endocytosis. They bud from membrane and they can enter by fusion of the envelope. Now, binding to the receptor is critical. And that's why only cells which express particular receptor for virus can be infected. And not every virus can infect every cell. They can only infect those cells which have receptor for, say, proteins or glycoproteins. That is what determine the viral tropism. And non-enveloped viruses, we know that enveloped viruses can fuse with target membrane, so viral envelope fuses with cell membrane. Non-enveloped viruses can enter by receptor-mediated endocytosis, and from endosomes, they can employ different mechanisms how to exit them. Some viruses trigger bursts of endosome and viruses are released. Another trigger hole in the endosomal membrane and they are released. Some exception, similar to bacteriophages, where virus actually drills a whole tunnel in the target membrane also exists. For example, picornaviruses, upon attachment to target cells, their fusion protein is open and fuses with the membrane and triggers the openings through which the viral RNA enters a cell. So there are many different ways of viral entry. And in addition to it, for example, some viruses such as pox viruses can use cellular mechanisms such as formation of philopodia, which would push them, those philopodia would push that virus from one cell to another, so they get from one cell to another with the help of the infected cells and actin rearrangement and formation of philopodia. Some other viruses, such as measles virus or HIV in macrophages, can form syncytia. So the virus exerts its fusion protein into the membrane of infected cells, and this can trigger fusion with other non-infected cells. So virus does not even need to be released from infected cell. It can actually be transmitted from via fusion. And these multinucleated giant cells are called syncytia. Viruses such as HIV also can form so-called virological synapses. You know about neurological synapses. In virological synapse, the envelope virus carry adhesion molecules on the surface which are derived from the infected cells, and this adhesion molecule will interact with another one on the target cells and foreclose synapse, where virus is directionally released into that synapse, so it does not have long way to reach the target cell and infect it. So there are many different ways in viral entry that can mediate transmission of virus from one cell to another. And what kind of receptor, what kind of cellular proteins can serve as a receptor? We know, I repeat again what we discussed before, that the receptor is that lock which needs to be opened by the key. And that key is protein or glycoprotein on the surface of virus. And if that key fit into the lock, the opening is granted. So virus can enter target cells. And there is difference between receptors and attachment factors. Sometimes, for example, there are a lot of chemokines on the surface, or there are a lot of glycoproteins on the surface which can interact with the surface of the virus. So virus binds to them temporarily, and this trigger concentration of the virus on the surface of cells, but those proteins or glycoproteins cannot mediate entry, so they are just attachment factor. Real receptor can mediate viral entry upon binding. And some viruses use just one receptor, for example, poliovirus receptor, which is immunoglobulin-like receptors. Measles virus use CD46, which is expressed on the activated lymphocyte. LDL receptor can be used by some rhinoviruses, for example. But other viruses require two receptors. For example, adenoviruses first binds to CAR, Coxsackie adenovirus receptor, which is also immunoglobulin-like receptor, and only upon binding CAR, it can bind to integrin alpha B beta 3-5, and only that one enables entry. HIV, the same way. Major receptor is CD4. This is for immunoglobulin-like domains. And this binding to first receptor trigger conformational change in GP120, which opens the binding site for co-receptor, which is chemokine receptor, and that one trigger further conformational change, which open fusion proteins. So receptors on the target cells can be very different. But what is a common characteristic is that most viruses use receptors which are highly conserved because of their function, so they cannot easily change to avoid viral binding. Now, in some cases, viral protein is not something which extend from the viruses, but it can be surface protrusion or canyon, such as in picornaviruses. Some viruses enter through cluttering-coated pit via endocytosis, and they are delivered into early endosome from where they can exit different way. Some viruses trigger micro or even macropinocytosis, so they trigger higher ruffling and invagination of the membrane, which afterwards non-specifically takes the material from outside into the cells. So that's how it enters into cytoplasm. Most viruses which enter by endocytosis requires lowering of pH as early endosome move inside of cytosol. We know that pH lowers, and this lower pH trigger changes in virus, which either trigger rupture of the vesicle or trigger hole in the endosomal membrane. So you see, there are full variety of different way how viruses can attach and enter into the target cells. And once they enter, once they bind to receptor or enter by endocytosis, they need to fuse. Envelope proteins have so-called fusion proteins. And this fusion, as mentioned before several times, can happen either on the cell membrane, so viral envelope fuse with membrane, cell membrane, or this fusion might happen in endosomes when virus is uptaken by endocytosis and at low pH trigger changes, conformational changes in the viral glycoproteins and opens the fusion proteins. So the fusion proteins are generally transmembrane protein, which can be class 1, 2, or 3. So class 1 are mostly made of alpha helices. This is pre-fusion stage, and on the bottom is post-fusion stage. In pre-fusion stage, you can see class 1 mostly alpha helices, which form trimers, and these trimers are perpendicular to the viral membrane. Class 2, for example, flaviviruses. I forgot to say class 1 influenza virus has this fusion protein. Flaviviruses have class 2 fusion protein, which are mostly composed of beta sheets. They form dimers, not trimers, dimers, and they lay parallel to the viral membrane. And this is after fusion, they completely change conformation. And recently, it was also described class 3 in vesicular stomatitis virus, G protein, and this is combined structure, which is defined by 5 different domains within those proteins. And basically, this trigger trimeric association. It is elongated fusion protein, and basically it has perpendicular, structure and also parallel structure. This is only recently discovered fusion protein, so we don't know as much about class 3 fusion protein as we do about class 1 and class 2. So how do these fusion proteins work? This is an example, for example, of influenza virus, which entered by endocytosis. And once the pH drops in the endosomes, this trigger conformational changes and viral fusion proteins, which are normally hidden, are changing conformation and now insert into the endosomal membrane. This can happen also in the plasma membrane, but in plasma membranes, the fusion is pH independent. Only in most viruses which enter via endocytosis, the fusion is pH dependent. So once those fusion protein are exposed, they are hydrophobic and they insert into the target membrane. And you see they are trimeric and several of them inserts. Once this insertion trigger further conformational change and they start to fold, and this brings these two membranes together, and as if this membrane fuse, it creates a channel through which the viral nucleic acid can be released. And here is it in the detailed mechanism. In a nice video, you see the fusion protein change conformation inserts to the target membrane. This insertion trigger additional conformation changes, and those fusion protein fold on itself and bring those two membranes together, and this create membrane to membrane fusion.